This is the science corridor linking many of the labs. We have an ice machine just down the corridor. It's just located here. Okay, so just spin around. So this is our prep room. And I'm just going to enter the lab. So this is from the science corridor. Okay, so this part of the lab, we call this a kind of our where we do most of our protein DNA work. So we have to the left is a biosafety cabinet, so we can grow bacteria. So we do a lot of our kind of aseptic technique here. We also have a shaking incubator, so it's floor standing, which will take various size flasks. I'll just spin round. We've got various like fridges. Here we have an ultracentrifuge. We've got a qPCR machine, real-time PCR machine. We've also got a PCR machine. Two types actually. We've got a gradient and also an ordinary PCR machine. See on the floor you've got two static incubators. We use one for growing fungi, so things like Saccharomyces cerevisiae. Also we've got second incubator for growing bacteria, and we can grow class 2 up to level 2 bacteria. We have also just on the end of the bench here is a plate reader. You can measure fluorescence, so we do a lot of cell variability assays using grisazerin. Okay, so we have a fridge freezer and also another fridge freezer next to it. We've got various kind of chemical stores. We like to keep the blue cabinets is used for keeping lab coats, so we like to have our own trays in the cupboard. And this is the door out into the office. We have a wash area, and then a spin round. In the corner here we have a ChemiDoc, and we can visualize DNA protein gels, and also it has fluorescence capacity as well, so that's quite a useful bit of equipment. Spin round. We also have a hybridizing oven, so if we want to do things like northern blots, southern blots, use this. Also for stripping western blots. Spin around to the bench here. We have a pre-cooled centrifuge, so microfuge, and then also we have a shaking platform. We also have a shaking therm mixer and various mixers, water baths. Just come around here. We also have this is called a DS11. It's a spectrophotometer fluorometer. So you can use this for it's a bit like a nanodrop. We run various protein gels. So we use these benches for running protein gels and also the DNA gels. Spin round. We also have Western blot, it's a semi-dry system. So if we want to do things like Western blots, we can use this system. And various spectrophotometers, more centrifuges, and also if we're interested to run 2D gels, this is it's an isoelectric focusing system from GE Healthcare. Okay, so we'll head on into the clean room. The clean room's separate to the main lab, so we like to keep this separate, and we normally change lab coats when we're working in this lab. We have liquid nitrogen storage, so we can mainly store cell lines in our Jura. We have two biosafety cabinets in here. These are laminar flow hoods, so we do like tissue culture work in here, so we can work with primary cells, or we mainly work with like cancer cell lines. We've got it's a tall fridge, some storage area. And then we have incubators. These are for cell lines. These are humidified with 5% carbon dioxide. And then we have benchtop. So it's like it's called a megafuge. So it takes Falcon tubes, goes up to about 5,000 RPM. and then another centrifuge. So we have quite a bit of bench space in here, which is quite good. We've moved some equipment around just recently. Okay, so what I'll do is I'll just kind of spin around the room, so give you an idea of the space. There we go. And we use these navy blue lab coats just for this room, so when we're working with tissue culture. Okay, so I'll head back into the main lab. Okay, and so, okay, so sorry, okay, so we have a prep room, so this is where we do all our sterilising. So we have a large autoclave that's mainly used for sterilising waste from the lab. We have a microwave, got all our glassware in here, weighing station, small autoclaves. So we use these for kind of autoclaving solutions, bottles, and then just spin around. So it's just the sink area, and then that kind of looks out into the science corridor. Okay, so head into the next part of the lab. So this is, the light will come on in a second. So this is it's really set up as a chemistry lab. The lights will come on now. There we go. So we have a still various fume cupboards. So we've got 6 fume cupboards in this lab, which is quite useful. And also we have the chemical storage underneath, so I like to keep those chemicals separate. What's useful in here for us is we've got an oven for drying tips. If we sterilise our tips, so we can dry them in this oven. There we go. So I mean, this lab space is being used for teaching. We have a community project with Pfizer, as this is kind of introducing science to local schools. Students who are not able to access laboratories, they're able to kind of acquire skills in this laboratory. This here is fantastic. It's a store. And it revolves, so when you open the door, you can actually revolve all the chemicals inside, so we use that as a store. There is a infrared IR just over here. That's really connected with the community lab. We could actually access that equipment. And also on the bench over there, we have, it's a small MMR. In campus we have, we've bought an MMR from Oxford Instruments. I can send you the details if you're interested. Anyway, that kind of concludes the tour. I'll just kind of give you a quick spin round again of this lab. There you go. Any questions, let me know.